Hello and welcome everyone. Yet another topic today, that's fatty liver and lipotropic factors. I've recently posted or published lipotropic factors as a single topic, and now I will present the same along with fatty liver. So, fatty liver, the term fatty liver represents excess deposition of fats in the liver beyond its capacity to utilize or transport them. So, when there is excess deposition of fats in the liver either does not have the capacity to utilize that quantity and neither is it able to transport them. Then, in that condition, the fat starts depositing in the hepatocytes and it's called fatty liver. The normal concentration of lipids in the liver is less than 5%, and lipids are actually synthesized by the liver but they are not stored by it. So liver is not the storage organ for fats, it's the adipose tissue. In normal liver cell Kupffer cells, the lipids are found in form of small droplets. Now let us look at the different types of fatty liver. So fatty liver is of two types, that is the non-alcoholic fatty liver disease and the alcoholic fatty liver disease. So we will be focusing on the non-alcoholic fatty liver disease in this presentation. So non-alcoholic fatty liver disease is commonly associated with metabolic syndrome, obesity, diabetes and hyperlipidemia. Let us see the further classification of non-alcoholic fatty liver disease. So the spectrum includes non-alcoholic fatty liver, which is abbreviated as NAFL. NAFL is defined as the presence of hepatic steatosis with no evidence of hepatocellular injury. So in such a condition, there is deposition of more than 5% or more than 5% of uh, lipids in the hepatocytes, but the injury to hepatocellular damage is not visible. The second kind is non-alcoholic steatohepatitis, that is abbreviated as NASH. And NASH is defined as the presence of hepatic steatosis and inflammation with hepatocyte injury, which is known as ballooning. So this is an advanced uh, case of non-alcoholic fatty liver. The third condition in this spectrum is known as cirrhosis, and in cirrhosis there's permanent scarring that damages the liver and interferes with its functioning. So this is a normal liver with lipid excess. There is non-alcoholic fatty liver disease, which is further advanced due to more accumulation of lipids, which causes inflammation and it leads to NASH, that is non-alcoholic steatohepatitis, which further advances to cirrhosis. So this is the complete spectrum of non-alcoholic fatty liver disease. Now let us see the difference between non-alcoholic fatty liver and NASH. So, non-alcoholic fatty liver disease is the most common liver disease. Fat accumulates in the hepatocytes, but there is no damage. High fat diet or uncontrolled diabetes mellitus are the most common causes. Non-alcoholic fatty liver disease includes both NAFL and NASH, that is NASH. So, non-alcoholic steatohepatitis, that is NASH, is an advanced form of non-alcoholic fatty liver disease. It is caused by progress of non-alcoholic fatty liver disease due to inflammatory reaction and further accumulation of fat. Cirrhosis. NASH can progress to liver cirrhosis and fibrosis. It can also go ahead with hepatocellular carcinoma with about 30 to 40% of patients. Fat molecules infiltrate in the cytoplasm of the cell. This is known as fatty infiltration. These are seen as large fat droplets in the cytoplasm. The nucleus is pushed to the side of the cell, which is known as karyorrhexis. Ultimately, the hepatic cell is lysed. As the healing process begins, the fibrous tissue is laid down, causing fibrosis of the liver, and further, as the fibrosis advances, there is scarring, and this is known as cirrhosis. So, let us look at the stages of liver damage. This is a healthy liver. Lipid does not store more than 5% of lipid fats. Accumulation of fat in Kupffer cells in form of droplets, but no or little inflammation. Followed by steatosis and inflammation. There is abnormal accumulation of fats in liver in steatosis. However, this is a reversible condition. With decreased lipids, the condition can be reversed and the liver can become healthy again. In steatosis, accumulation of fat is accompanied with liver inflammation, fiber tissue is formed eventually, and with that, it causes fibrosis. In fibrosis, there is, as it advances, there is scarring in the liver, and this is causing healthy tissue of liver to become scarred due to chronic inflammation. So fibrosis is the first stage of liver scarring. However, fibrosis is also, in the early stages, it is reversible, and with decrease in lipid levels, it can be reversed to a healthy liver. As the healthy tissue of liver becomes scarred due to chronic inflammation, the fibrosis advances and the scarring is there, and this leads to cirrhosis. Cirrhosis cannot be reversed, and the first three stages are generally reversible. The scar tissue replaces the healthy liver tissue and it's permanently damaged, so it cannot be reversed. And from here, now, as with the advancement, the liver undergoes liver failure, so the only option now here remains is to have a liver transplant. A cirrhosed liver can undergo blockage in the circulation by scar tissue, and that causes liver failure, so a transplant becomes a necessity. Otherwise, the person may will lose life. The cirrhosed liver can also advance towards hepatocellular carcinoma, and these conditions are all irreversible. So these are the stages of liver damage. So now let us look at the biochemical causes of fatty liver. The biochemical causes of fatty liver includes fat deposition in liver, and this may be because of increased triacyl glycerol synthesis. This is the first category which leads to fatty liver. The causes are excessive mobilization of non-esterified fatty acids from adipose tissue, and the second cause is excess synthesis of fatty acids from the glucose. So these are the two causes which lead to increased triacylglycerol synthesis and which causes fat deposition in the liver. The second cause which leads to fatty liver is reduction in removal of fat from the liver, and the causes include toxic injury to liver, because of which certain apoproteins like ApoB100 and ApoC, which are essential for VLDL synthesis, are not synthesized. There is also decreased oxidation of fat by hepatic cells, and this also causes fat deposition in the liver. Now, even because of hepatitis B, there may be hepatocellular injury, and because of which the VLDL synthesis will be defective, and so removal of fat from liver will be slow. So increase in triacylglycerol in the liver due to factors 1 and 2, or decrease in removal of triacylglycerol due to factors 3 and 4 cause excessive fat accumulation leading to fatty liver. Now, the deposition of excess triacylglycerol in liver. Let us look at the detailed biochemical explanation for deposition of excess triacylglycerol in liver. So, there is increased triacylglycerol synthesis. The factors that cause this increase is high calorie intake, diabetes mellitus, and starvation. So, when there is diabetes mellitus or starvation, the glucose is not available for energy and therefore fat is mobilized from adipose tissue. There is breakdown of triacylglycerol. The free fatty acids or the non-esterified fatty acids are circulating in large amounts and they reach in large amount in the liver. So when these free fatty acids reach the liver, they are again uh, esterified to form triacylglycerol. Now, what happens is that the liver has its capacity to synthesize VLDL. So, the capacity of liver to take up fatty acids from the blood far exceeds its capacity for excretion as VLDL, and therefore fatty liver can occur in diabetes mellitus and starvation due to increased lipolysis in the adipose tissue. So let us look at the reasons why there is reduced utilization and reduced removal of fat from the liver. Let us see the detailed biochemical explanation. The most common cause for this is chronic alcoholism, and as you know that ethanol is metabolized to acetaldehyde by alcohol dehydrogenase, and acetaldehyde is further metabolized to acetate by aldehyde dehydrogenase. And in both these reactions, NADH is formed. So NAD plus is reduced to give NADH in the first reaction as well as in the second reaction. Because of this, there is increased ratio of NADH to NAD, and for this reason malate is no longer available for the synthesis of oxaloacetate. So when there is decreased oxaloacetate, there is a blockage in the TCA cycle, and so due to fatty acid oxidation, whatever acetyl CoA is obtained, it cannot enter the TCA cycle, so there is reduced utilization of fat. Now because of this, the fatty acid oxidation is blocked, and so this increased fatty acid will now be synthesized to triacylglycerol, and this increased triacylglycerol is so large in a number that it exceeds the capacity of the liver to remove this TAG from the liver in form of VLDL. So this is the second cause. Now let us look at the third cause, that cause biochemical cause for fatty liver. It is decreased VLDL synthesis or its decreased secretion. Now this is categorized into uh, decreased VLDL synthesis or decreased VLDL secretion. Let us look at the causes of decreased VLDL synthesis. It can be due to decreased apoprotein synthesis. The decreased apoprotein synthesis may be due to protein energy malnutrition, which leads to decreased amino acids, and certain amino acids are needed for the synthesis of ApoB and ApoC. ApoB and ApoC are required for VLDL synthesis. So if there is protein energy malnutrition, amino acids will be deficient. So ApoB, ApoC synthesis will not occur. So there will be decreased VLDL synthesis. So due to decreased VLDL synthesis, the fat will deposit in the liver. Now, the second cause of decreased apoprotein synthesis is hepatocellular injury. The hepatocellular injury may be caused by viral hepatitis B or uh, due to some toxic compounds like carbon tetrachloride, arsenic, lead, or ethionine. All these injure the hepatic cells, and because of the injury, uh, the apoprotein synthesis and VLDL synthesis are affected. So there is accumulation of fat in the liver. Now, the second reason for decreased VLDL synthesis is decreased phospholipid synthesis, and this decreased phospholipid synthesis is uh, because of decreased choline, inositol, and decreased free fatty acid availability. So, all these three are required for phospholipid synthesis, and phospholipid synthesis is essential for VLDL formation. So, if these are not available, there will be decreased phospholipid, which will lead to decreased VLDL, and so decreased VLDL will allow the fat to get deposited in the liver. The fourth cause for VLDL synthesis decrease is decrease in essential fatty acids and increased cholesterol intake. Both are essential for phospholipid synthesis. Now we go to the uh, second category or second cause of decreased VLDL synthesis, that is decreased VLDL secretion. That is due to deficient lipotropic factors. The lipotropic factors include choline, inositol, methionine, betaine, vitamin E, selenium, omega-3 fatty acids. How these lipotropic factors uh, prevent the uh, uh, accumulation of fat in the liver, we will see in the next coming slides. The mechanism of lipotropic factors. So let us see how these lipotropic factors work. Lipotropic factors are basically required for removal of normal mobilization of fat from the liver. They prevent accumulation of fats in the liver, and this phenomenon is known as lipotropism. The dietary deficiency of these factors can result in fatty liver. So, choline and inositol are two examples. The second group of examples include methionine and betaine. Let us see their mechanism of protecting the accumulation, protecting the hepatocytes from accumulation of triacylglycerol. So, choline is a principal lipotropic factor of our body. It's a constituent of phospholipids, and the choline is responsible for forming phosphatidyl choline, which is also known as lecithin, and inositol is needed for the formation of phosphatidyl inositol. Both are integral components of lipoprotein VLDL. Now, when there is sufficient amount of choline and inositol, then lecithin and phosphatidyl inositol will form, and that will lead to VLDL synthesis. When VLDLs are synthesized, they will export all the triacylglycerol from the hepatocyte to the peripheral tissue. Methionine and betaine are methyl donors. They are required for the synthesis or formation of lecithin, which is a constituent of phospholipid. Methionine acts as S-adenosyl methionine. It's a methyl group donor for the formation of choline. Choline is required for the formation of lecithin. It is also required for the synthesis of carnitine. Carnitine is a uh, s- transporter for fatty acids for the oxidation of fats, and it is synthesized from lysine and methionine. So, when choline and inositol are deficient, then there will be decreased phosphatidylcholine. So, there will be decreased VLDL synthesis, and that will lead to decreased triacylglycerol export, causing accumulation of lipids in the fat in the liver, leading to fatty liver. When methionine and betaine are low in quantity in our body, the formation of lecithin will decrease, and that will lead to decreased formation of choline, and it will also lead to decreased carnitine synthesis, therefore decreased oxidation of fats. And all these will again lead to accumulation of fat in the liver, and which will eventually lead to fatty liver. The third group of lipotropic factors include vitamin E and selenium, and the fourth group includes vitamin B12 and folic acid, whereas the fifth group includes essential fatty acids. Now, vitamin E and selenium are antioxidants, and they prevent the oxidation of unsaturated fatty acids and lipids, and therefore they prevent oxidative stress. So, when vitamin E and selenium are deficient, there is increased oxidative stress in the body, which further causes injury to the hepatocytes, which are laden with lipids. B12 and folic acid are needed for the formation of methionine and homocysteine metabolism, and when B12 and folic acid are deficient, methionine synthesis will decrease, which will lead to decreased choline synthesis, and that will further lead to decreased phospholipid synthesis, which are necessary for VLDL synthesis and triacylglycerol export. Essential fatty acids are required for formation of phospholipids. So, for example, arachidonic acid. So, the protective effect uh, of essential fatty acids is given by the essential fatty acid because they will form phospholipids, which will in turn be responsible for VLDL synthesis and export of triacylglycerol from the liver. If essential fatty acids are low in the diet, then it will lead to decreased export of triacylglycerol from the liver. All these factors will cause fatty liver. So I hope by now you know that there are different lipotropic factors. The most important lipotropic factor or the principal lipotropic factor is choline, and these factors are necessary for prevention of fatty liver. So they protect the liver by facilitating removal of triacylglycerol from the liver. It is done by two processes. One is by synthesis of VLDL, and second is by synthesis of phospholipid choline. So these are the phospholipid for which choline is necessary. So thank you for watching my video. Like, share and subscribe, and keep watching my videos.